I welcome your comments and your feedback about things that worked in Fall 1 for you and things that didn't work in Fall 1 for you. We're going to go through my plans for this semester, this term, pretty soon. But I welcome your input. And I might not do what you suggest, but I'm happy to incorporate your ideas. So let's, before I go on, let me make an announcement. I'll probably do this pretty much every class. This is not related to why is the size of it? Oh, we'll do it that way. So Brent Smith is offering a AWPE course in trail building and maintenance. So if you like to be outdoors and you would like to get a AWPE credit and you would like to work on Saturday afternoon to get your AWPE credit, contact Brent Smith. So let's look at the PowerPoint. You're juniors and seniors, you know the drill. How many times have you heard a professor say, please read the syllabus, there's information in the syllabus that's important? Well, I just said it again. So because there is information in the syllabus that is important, we'll talk about some of it, but we're not going to go through the syllabus. You know how to read, you know how to incorporate information. Please read the syllabus, and if you have questions about the goals or the grading rubric, the grading, the course value, the points, please contact. So I'm going through my stack of stuff. So hopefully you all picked up the papers here. I provided handouts for things like the syllabus. So there's the syllabus, which I ask you to read once again. There's the course schedule. So the course schedule is the table. It looks like this. There are two versions of it. One has the reading assignments for the 13th edition of the textbook, Vander volume, 13 edition. That's the one that, if you took AMP 1, you probably already have Vander 13. If you didn't take AMP 1, you have a choice to buy either the 13th edition, which is now 5 or 6 years old, or the 15th edition. 14 I skipped. So the 15th edition is the newest edition. You can either use 13 or 15. Either one is fine with me. If you have the 13th edition, you have the schedule with the page assignments for the 13th edition. If you have the 15th edition, I have a revised schedule here with revised page numbers because the pages are different, that I will give you. So if you have or plan to get the 15th, the newest edition of the physiology textbook, please come and get this schedule. I didn't put them both there because I thought it would be confusing. So the schedule. So you'll notice that we have a fairly standardized weekly schedule. I'm a pretty strong believer in pattern recognition, and so I'm trying to set up a pattern, weekly pattern. So let's discuss. Let me just tell you what my plan is for the weekly pattern. I emailed this to you, but I know it was during break and you may not have studied your email. So on Mondays, like we are right now, we're going to have a lecture, an interactive lecture. You probably know that my style for lecturing is not just to talk, but to try to engage in a conversation using clickers and just, think, pair, shares. Well, we won't be doing that, but 1 minute writes. So an interactive lecture for about an hour, maybe an hour and 20 minutes, probably with a break in there somewhere, just to get up and move around. Then a longer break, maybe a 10 or 15 minute break. Then a period during which you will be here working individually on problems that I will assign you, either problems from the problem set that is due later that week, or additional problems that I think are educational. So I'm under some tension here, right? On the one hand, I want to do the best job I can do in being an educator, and on the other hand, I need to keep you safe, and I want to keep myself safe. So the educator part of me says, let's do this in person, because I heard words from colleagues and from students that this was better for education. From a safety standpoint, I would love to have you work together, but I can't. I can't have you sitting next close to each other, working on a problem together. So what I'm going to do, what I'm planning to do is on Mondays, you will work individually on these problems for about a half an hour. And then we'll have another lecture. On Wednesdays, the schedule will be similar. We'll start in here, interactive lecture, break, during which you will scatter to your favorite Zoom location, and then you will come to class via Zoom. I will break you out into breakout rooms where you will work together on the same problems or on additional problems via breakout rooms, working together, but safely via Zoom. I think this will, I hope this will work. This will allow you to share notes. I'm a pretty strong believer in working together is good for everybody. That the students who benefit from hearing the information from another student get that experience in a working together scenario. The students who get it better by explaining it to somebody else. So it's good for everybody. And so we will work together, you will work together in breakout rooms on Wednesdays. I'll be available, in the main Zoom room, to be asked questions. I'll be right there. So today, when we break and I ask you to start working on the problem set after my first hour of lecture, you'll be sitting here working. I'll be standing right here. If you have a question, you just come up and ask me a question. What do you mean by this? Is this the right idea? Da, 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 da. On Wednesday, it'll be the same idea, except that I'll be in a Zoom room, and you'll be in a Zoom room with some other students doing the same sort of thing together. And then coming back to me to ask me a question. And then we will have, after that breakout room work, working together on problems, we'll have a Zoom lecture. So you won't come back here for the follow-up lecture. It'll just be on Zoom, because once you're scattered, you stay scattered, right? So there'll be 3 lectures a week in this room: Monday 1, Monday 2, and Wednesday 1, and then 1 Zoom lecture each week. The Wednesday part 2 will be on Zoom. All of these will be recorded. So Brittany and Joey, who I'm sure you know, will be recording this room, and the Zoom lectures will be recorded on Zoom, and they'll all be posted either on YouTube or on Kaltura or somewhere. I'll figure that out. The recordings are for your review. They're a resource for your review. If you miss a class, they're a resource for not missing the class completely. So that's Monday and Wednesdays. On Thursdays, I've blocked out the whole day for office hours. So I'm not going to be sitting at a Zoom room waiting for you to log in. I may not even be sitting in my office, literally, waiting for you to come to office hours. But I'm making myself available all day Thursday. So all you've got to do is send me an email and say, can we get together for 15 minutes? Or so-and-so and so-and-so and so-and-so and I want to get together with you as a group. Can we set up a Zoom meeting together? So they can be in my office, they can be on Zoom. whatever you want, but you just need to contact me and say, I would like to have an office hour meeting with you. It's not limited to Thursdays. You can try to do that on a Tuesday or a Wednesday or a Friday, but my time is more cut up by other things on those days. My time is available almost all day Thursday. So Thursday night is when your problem set is going to be due. So you have the problem set now, so the deal is on Mondays you will get the problem set and work on it individually on Mondays, work on it at home on Tuesdays, work on it in a group with other students on Wednesdays on a Zoom breakout, turn it in Thursday night. My hope is that you will all get 100% on all of the problem sets. If you work on it together and individually together, there's no reason why you shouldn't get a perfect grade on a problem set. Well, of course there's a good reason why you should not get a perfect grade. If you don't do, if you don't invest 100% of your effort, which I understand, you might not get 100% on the problem set. But if you do invest full effort, it should be really easy to get a 99% or 100% on problem sets. I'm not saying they're easy problem sets. I'm saying that with the amount of work that I'm asking you to do, you should be able to do very well on problem sets. So Thursday night, the problem sets are due. In the past, I've set up an envelope at the CST desk where you would turn in your problem set there. I'm going to do that, but the CST desk closes at 9 p.m. So if you are it's due Thursday night. If you miss the 9 p.m. deadline, you slide it under my office door. If I get it Friday morning, and I'll come in early Friday morning, I'm an early bird, so I'll get it early Friday morning, and it counts. So if you turn it in at 4 a.m. on Thursday night, I won't know whether it was 4 a.m. or 11 p.m. the night before, before. It's fine. That's your backup. But it's got to be there by the time I get it Friday morning, either in the envelope at the CST desk or under my office door. If it's not there, it's late, but still turn it in. Even if it's late, turn it in, it's a late penalty. 10% a day is the late penalty, including the weekend. So if you turn it on Saturday, the best you can do is 90%.
Are the problem sets posted on Moodle so we can work on it on a computer?
Yes.
Okay.
Yes, the problem set is already posted on Moodle.
Okay, so there won't be any because if we don't want to come in here to turn it in, there won't be any online turn in.
If you have an electronic version and you want to just send me a email attachment,
okay.
Let's do it that way.
Okay.
I think for many of these problem sets, writing it out will be helpful. It might, some of it will require some calculations and typing formula and equations. It's just cumbersome.
Okay.
So for example, on this problem set, there's some arithmetic that you have to do. So it might be awkward to try to type that into your problem set. But if you could print it out, or take this hard copy, write it out, scan it, and send me the scan. I'm willing to receive electronic versions, and if a lot of you are doing that, I'll set something up on Moodle for an electronic depository for problem sets.
Okay, thank you.
Make sure that if you do send me an email, your name is on the document, and preferably your name is also in the file name. Is that okay? So now we're going to get to Thursday night. Thursday night, I would like to have a lab option. This class is a little too big for one lab to do safely. So I would like to know how many of you could, even if you don't prefer it, but how many of you could, in principle, do a lab on Thursday evening from 7 to 9:30 or so. Okay, good. So, now just out of curiosity, I'm not going to be remembering any of these hands right now. How many of you might even prefer a Thursday evening 7 to 9:30 option? Okay, that'll work. So we're gonna do this in a few minutes. So Thursday evenings, some of you will have lab, and all of you will be turning in your problem set. Friday, we will not have lecture. Friday is a lab day, so if you're in the Thursday lab, you don't have anything on Friday. And if you're on the Friday lab, you'll come to lab at 1 p.m. on Friday for lab. The lab is going to be a mixture of anatomy, human anatomy. I'm sure you've heard about this course. You'll be working with the cadaver. And so when you come to lab, so I'm guessing there's going to be like 8 students in each lab section. And then I'll probably divide that 8 students into halves. So one half of you will be working out in the main lab doing something anatomy related. And one half of you, 4 students, will be in the cadaver lab. And we'll do that for about an hour, maybe an hour and 15 minutes, and then we'll switch. And so, for example, this week, out in the main anatomy lab, you'll be doing anatomy of pig hearts. And in the cadaver lab, you'll be working on the cadaver heart, the human heart. Well, I'm getting ahead of myself. That's what that Thursday night or Friday afternoon will be anatomy lab. There'll be some physiology experiments in there too. On Friday at about noon, I'm going to make a quiz available at the CST desk, and it's due on Sunday night. This is closed book. You can't work with your friends. You can't open the internet. You pick up the quiz, you sit down, you do it, and you turn it in. This one can't be done electronically. This one you really have to come to CST and pick it up and do it. If there's a reason, like if you get quarantined and you can't do that, I'll make arrangements. But if you're not in quarantine or isolation, and I'm not sick, that we'll do it at the CST desk. So you go to the CST desk, you pick it up, and you have to be mindful of the CST desk hours, which I've written down here somewhere, but you'll find out the hours. Like on Friday, they close early. On Saturday, they're open noon to 4, and on Sunday, they're open noon to 9. So again, you pick up the quiz, you sit down, you take it, closed book, leave your book somewhere else, preferably leave your phone somewhere else. Avoid the temptation, because looking something up is cheating. And, cheating is a form of lying, and I won't lie to you, so I'm asking you not to lie to me. So for example, if you go to the CST desk at 8:45 p.m. and on Sunday, and you're still working on it, and the desk is closing, and you can't turn it in at the envelope at the desk, you bring it to my office and you slide it into my door. But you can't pick it up after 9 p.m. on Sunday. That's your deadline for getting it. So that's your deadline for starting it, because I want you to, when you pick it up, I want you to not take it out of the building. It should not leave the building. But the building's CST and Stanford. Okay? Any questions about so the quiz is due Sunday night, and we're done with the week. We'll start again on Monday the next day. Any questions? Brittany?
I just wanted to add that on Mondays, I do drop-in tutoring over Zoom if you guys go to the Academic Enrichment Center. So that'll be every single Monday. And then if you guys have any questions outside of that time, I am also available for one-on-one tutoring.
Okay, good. So we've got, so in addition to my office hours on Thursday, you have Brittany's office hours on Monday, right? Did you just? drop in hours on, specifically on Monday?
Monday nights, yeah, 7 to 9.
Okay, so let's, any other questions?
Well, when did you say the quiz will be available?
The quiz will be available on Friday. I'll try to make it available early on Friday. I promise to make it available by noon on Friday. If you pick it up on Friday at 3:45 and the desk closes and you're still working on it, keep working on it, do the quiz and slide it under my office door. You might even write, the date and the time on it. Because when you sign out the quiz from the CST desk, there's a time stamp on it. And when you turn it in, there's a time, they mark the time. But if you don't turn it in there, you turn it in under my office door, just write the time, just so I know. This is unusual to be asking you to slide it under my office door because, the CST desk hours are just not really compatible with what I know your schedule would be, to be working on it Sunday night and be done with it, let's say midnight, so you could get a good night's sleep, but still work on it Sunday night. 9 p.m. just seemed too early. So that's why I'm offering the option to slide it under my door.
Is there like a time limit for them? Or
There is no time limit. I think they can be done in an hour or less. I think you should plan on an hour. Brittany, you think that's reasonable?
That's definitely fair. I'm a fast test taker, but I used to finish them in like half an hour. So yeah, I would say an hour is good.
Yeah. Those of you who took AMP1, the quizzes are going to be similar in size, not in content, but in size. And so if it took you half an hour to do an AMP1 quiz, it'll take half an hour to do one of these. And if it takes you an hour to do an AMP1 quiz, it'll take you an hour to do that. If you're fast, it'll take you half an hour, and if you're very careful and methodical, it might take you an hour. Okay, let's go through the PowerPoint just to see if I hadn't already said something. So AMP2 is covering the heart, the lungs, the kidneys, and the digestive tract. If you're interested in brain, neurons, muscle, that's AMP1. Course goals. Lots of content. Let's face it, one of my goals is for you to learn a lot of biology, the anatomy and physiology of those systems, the cardiovascular, respiratory, urinary, and digestive systems. And I'm going to ask you to think and make connections between the systems. Take the information you know about cells from Cell Phys, and even if I didn't explicitly teach it, apply it to the organs that you're learning about in AMP. I hope, even with COVID, distancing, I hope that you will develop teamwork skills and learning from each other. And, like I said, teachers, you probably have heard this a zillion times already, there's no better way to understand something than to teach it. So if you can teach somebody else your understanding of something, it's in your brain in a sort of in a way that is much more solid than if you just feel like, yeah, I understand it. Because if you say, yeah, I get that, let me move on to the next thing, and then someone says, can you explain it to me? And you can't. You didn't understand it as well as you thought you did. So this is the 13th edition of the textbook. It's required, or the 15th. Being able to read the textbook is something you are going to have to do. The reading assignments are required. They supplement what I'm going to be teaching in lecture. I think this textbook's pretty good. There's some of the figures in it are great. The difference between the 13th and the 15th edition are not major, so either one works fine. The coloring book is also required. I'm not going to check your coloring. I'm not going to see, oh, you used the wrong color here. I'm not going to do that. I'm not going to check how well you stayed within the lines. But I think it will be enjoyable to you to you know, learn anatomy through this process of coloring. I mean, it's just sort of, hopefully it's a little bit fun, and it's a little bit active, and it will help you learn the anatomy words by coloring the words and then coloring the structure. It's not a kid's book. Medical students use this book to help reinforce what they've learned in a really hardcore anatomy lab. So I think it's a valid way to learn anatomy. Again, I'm not going to be checking your coloring, but quizzes, especially the anatomy quizzes, and some of the questions on the exams could come straight out of the coloring book. So it's in your best interest to study from the coloring book. Those of you who took A&P 1 know that in this room, we're mostly going to be talking about physiology with a little bit of anatomy. And in the lab, we're mostly going to be talking about anatomy with a little bit of physiology. Questions about the reading or coloring assignments? They're in the schedule.
With the exams, we should expect questions from the reading and the PowerPoints, right? Or should we focus, because I know for A&P 1, most of the questions were stuff that we talked about in class on the PowerPoints. Like the reading was supplemental, but the focus was on the PowerPoints.
Yeah, let's be clear. What I teach in this room is going to be the most important material from there's the total content, and then there's the stuff in the content that I teach. And I will examine your knowledge of the most important content. I am not going to examine peripheral information. So the PowerPoints and your lecture notes are your primary source of information. And the textbook is, I think, an essential reference source. That's what I think. So I put up the at least part of the schedule. So you see, here's where we are now. Here is the reading assignment that is due today. Obviously, it's not due today, but this is all review material from self-in. So these are the pages that are going to be covered in these classes. So this is your reference to the pages in the textbook. And this is the 13th edition, but the 15th edition pages are a little different. Coloring book pages 1 and 2 are assigned. Ordinarily, so for example, on Wednesday, October 7th, I'm going to pretty much assume that you've at least skimmed the textbook reading, and you may already have done the anatomy coloring. I think you'll learn better in this room if you have already sort of planted some seeds of knowledge, or even planted some seeds of, I don't get this, and if Bob doesn't explain it, I better ask a question. I think you'll learn better here from me if you've done some homework before class, and then use this as a reference after class as well. Same with the coloring book assignments. Here's the problem set that's due on Thursday. So it's listed with the second lecture. That's funny. So this is Monday. Here's the problems that due on Thursday of that week. Let's just scroll down. So more quizzes and problem sets. Quiz due, problem set, quiz, problem set. So it's all there sort of in a pattern until we get to exam 1. So in the past, I've had an exam for each section. But that would mean putting an exam after about a week and a half of class. And I felt like, oh, that's a little fast. So this is going to be a big exam. It's covering 2 sections, 2 organ systems. So you want to keep up with the material. So the first exam is coming up in, 3 weeks from today. So you have three weeks of material. At the pace that we're going to go through is going to go real fast. So you want to keep up with it. Do not fall behind. We'll have an in-class exam. Then we'll get back to our schedule, quiz, problem set, reading, coloring. And then about 2 weeks later, we'll have another exam. And then we'll be back sort of on our schedule. And then at the end, we will have a lab practical quiz and a final exam. A cumulative final exam. So it'll be both the section exam for the final section of the course and the cumulative final.
Is the practical quiz just for the digestive system, or like everything we've learned?
Everything in the anatomy lab. So do you all know what a practical is? So I'll tell you when we're in lab, but in a lab practical, what I'm going to do is, you'll have seen the human heart, and you'll have seen the human lungs, and things will have gotten taken out of the body. And in a lab practical, I will pin various sections of the heart, the lungs, the digestive tract, the kidneys, with a little numbered pin. And you will write the name of that structure on an answer sheet. We'll go through that more in lab. Yeah?
Do you have a time on the final exam?
Time. I think it's class time, but I'm not sure. Is this is a Tuesday? Okay, so I don't know. I'll have to find that out. Let's see, where was it? Quizzes, problem sets. Okay, so we've already done all of this. We've talked about problem sets and the schedule. So I think I printed this slide out for you on your handout. So you've now got information in your head from me talking, in your syllabus, and now on your slide printout. The lab will be working on a human cadaver, the chest and the abdomen, because that's where the heart, the lungs, the kidney, and the digestive tract are. I can show you the work that was done on dissecting muscles that was done in the spring of 2020. But it's experiential learning. The point is to have the experience of dissecting a human body. It's amazing. This is a very famous painting by Rembrandt. And so you are going to become, some of you are already part of what used to be a brotherhood, but now is a community of anatomists. You have joined or will join this community of anatomists, and having had the experience of learning anatomy from a donor. I can't be sure that this person actually donated his body to this doctor. It's quite possible he had no choice. But that's not true of the body that we have upstairs. That person decided to give her body to us to study and learn from. We'll talk more about that in lab. So these are the systems we're talking about. Let's see, so this slide is my transition to material. But I want to break this, I want to do the lab section while we're all relatively fresh. So, why is it? I'll have to figure this out. Okay, so you should find your name on this list. If your name is not on this list, now is the time to let me know, because that'll mess up our partition. Is there anybody on this list who you suspect or have heard is planning to drop the course? So for example, Austin's not here, but he contacted me. Does anyone know? Oh, Ruthie's not here, but she contacted me. Sophia? Yeah, okay. Evan, does anyone know if Evan's planning to take the course? Anybody know one way or the other? Okay, well, I'm going to partition him into a section, but he didn't tell me which one he preferred. So what I'm going to do is just take, is everybody in this room on this list? Okay, so there's our list. So you can see, CJ gets to go first and Evan goes last. Okay, that was convenient. And so what I'm going to ask you to do is tell me your preference. Thursday evening or Friday afternoon? That'll do it.
Okay, I didn't know if you wanted me to tally the,
Oh yeah, if you could keep track of, yeah, that would be helpful. Keeping track, thank you very much. Keeping track of how many we have in each section. Okay, CJ, what's your preference?
Friday.
Okay. Sophie.
Friday, please.
Sheldon?
Thursday?
Robbie?
Friday.
Meg?
Oh, see Friday.
Couldn't hear you.
Friday.
Friday? Katie?
Thursday.
Easton?
I'll go Thursday.
Drenusha?
Thursday.
Mike?
I'll go Thursday.
Abby?
Friday?
Remy?
Thursday.
Andrew?
Friday.
Ruthie? What did she tell me? She prefers Friday. No. What's our count?
77.
Olivia?
Friday, please.
Friday? Austin? What did he say? Oh, he said Friday. No, he said Thursday night. Thursday night is good for him. Austin is Thursday. Sarah?
Friday.
So what are we at?
Evan has to go in Thursday. We're at, even
9 on Friday?
and 8 on Thursday.
Okay, so we'll put Evan on Thursday. Okay, so I'm gonna read these down to make sure that I got it right, so I wanna double check. I'm just gonna do this in alphabetical order. Andrew on Friday. Let's yell out, shout out if I got it wrong, please. Andrew, Friday afternoon. Austin, Thursday night. Sarah, Friday afternoon. Easton, Thursday night. Remy, Thursday night. Evan, Thursday night. CJ, Friday afternoon. Jernusha, Thursday night. Olivia, Friday afternoon. Sheldon, Thursday night. Michael, Thursday night. Meg, Friday. Ruthie, Friday. Sophia, Friday. Robbie, Friday. Katie, Thursday. Abby, Friday. Nahum, Thursday. Did I get it right? Okay. That's the plan. So now you know which, you have your section. So on Thursday, plan to come to lab. Those of you in the Thursday section, plan to come to lab at 7 p.m. sharp. We start on time. Even being a minute late is a penalty. We'll come on time. And then on Friday afternoon, let's say we'll start at 12:45, just to be easy about it. 12:45 on Friday afternoon. Okay. Just always I'm worried about whether it will work, and it always works. And we didn't even have to pull any strings this time. Sometimes I have to incentivize people switching. Didn't have to do that. Okay, so I'm going to stop discussing the course and start my interactive lecture on material. So you'll notice one of your handouts was a key concepts list. This is a study guide when you're learning the material, when you're reviewing the material for an exam especially. It doesn't have everything as, it's not a narration of what I'm going to say, but it has the key points. And if you say, boy, I don't remember him saying anything about calcium ATPase, you go back to your notes or you go back to the slide and say, oh yeah, there's what he was talking about with the calcium ATPase. So the calcium ATPase shows up, but it's just a name on a list. And you have to say, okay, well, what were the key concepts associated with the calcium ATPase point? So we're going to talk now about ions. Oops. We're going to talk about ions. We're going to talk about ion transport and other mechanisms, well, the mechanisms of solute transport. That's what we're going to do now for about half an hour, 45 minutes. Then we'll take a break. Then you'll start to work on the problem set. And then we'll come back and start talking. We'll be in the second lecture. And we'll talk about these other things. So this is going to go, this is going twice as fast as I'm used to teaching. But you're used to it. So this is review from Cell Phys. So this is even a slide that you saw in Cell Phys. Now I know for some of you it's been 2 years since you saw this slide in Cell Phys, so that's why we're going to review it. So here's the lipid bilayer. You remember the lipid has a hydrophobic tail and a hydrophilic head, and so if we were to cartoon the lipid bilayer, there'd be head groups out here and there'd be hydrophobic tails in here. You could even cartoon them into your slide to remind you that the lipid bilayer has a hydrophobic core and a polar surface on both sides. And membrane proteins are embedded in this lipid bilayer. Some of the molecules or ions that we are interested in this course are actually amphipathic enough that they simply diffuse both through water and through the hydrophobic core of a bilayer. Some of them do that, and we'll talk about that in the next slide. Most of the ions that we're interested in, sodium, potassium, calcium, chloride, phosphate, most of those ions cannot pass through a membrane, and so they have specific transport molecules. And we're going to put them into really 4 groups. Channels, which are a form of passive transport. Uniporters, which are another form of passive transport. And active transporters, and we're going to be talking about primary and secondary active transport. I don't want to reteach self-phys. I know that some of this is not in your brain anymore, so it's your job to go get it in your brain from either rereading your self-phys notes or reading the textbook. Okay? I want to be clear. One of the things that I should have mentioned when I was talking about the syllabus is that because this is a 4-credit course crunched into half the time of a usual 4-credit course, your job, your homework expectations are probably twice what you're used to having in a regular semester. So my expectations are that you will allocate at least 3 hours a day. Whether you use all 3 hours a day to study for the course is up to you. But I think this course needs, in some cases wants, some cases only benefits from, but let's just say needs, for you to spend about 3 hours a day on homework. So one of your jobs is to make sure you remember the concepts here. I'm going to teach it a little bit, but I have to move fast. You have more time at home than we have in this room. And so your job is to fill in the gaps of your knowledge from the reading, from the internet, whatever source you want. I'm happy to answer questions. And now, or on Thursday, or whenever. But the point I'm trying to make is that I won't teach everything that you need to know. Some of it will be, you'll have to figure out sort of on your own. I plan to teach the things that you haven't already had that are important. But this is something that's important that you've already had, and so that's why I'm going to move fast through. Okay, so some of the molecules that we're interested in this course, oxygen and CO2 in particular, especially in the respiratory system. Oxygen and carbon dioxide are both amphipathic enough that they dissolve in fluids and they can pass through lipid bilayers. So when you get to the lungs, you're going to learn about passive transport of oxygen and CO2 through the cell membranes in the lungs. Passive, not just passive transport, simple diffusion. Most of the other things that we're interested in this course, like amino acids and glucose in the digestive tract and in the kidney, ions in muscle and heart and kidney, they cannot undergo simple diffusion through membranes. So let's talk about how these ions or molecules get through membranes. There are uniporters and ion channels that both catalyze passive transport of these solutes that cannot undergo simple diffusion. It's passive. What do I mean by passive? it means the solute is moving from a high concentration to a low concentration. This is not active, so this does not set up a gradient. It, in fact, breaks down a gradient. The ion is moving, or the solute is moving from high concentration to low concentration, passively. Just like here, oxygen and CO2 are moving passively from high concentration to low concentration. You can't have passive movement from low to high. That's not a passive process. So let's see, what happens here? High concentration, low concentration. Let's just say, it doesn't say it, but let's just set it up. The transporter, you may notice, has a conformational change. From outward facing to inward facing, or from, facing this solution, let's just say this is the outside, facing this solution, to conformational change, facing the solute binding sites are now facing this solution. So a solute can bind, binds to the outward facing conformation. The ions can move within the protein. Conformational change, that's what this arrow is, to an inward-facing conformation, and then the solute could dissociate. And so binding, conformational change, unbinding allows for passive transport of the solute. One solute, one conformational change. It's one at a time. Solute binds, conformational change, movement, unbinding. Conformational change, back to the original conformation, binding, conformational change, unbinding. One movement of the solute per conformational change. Ion channels, let's go to the next slide. Ion channels also have conformational changes. They go from closed to open conformations. This is protein conformational change. Here's the lipid bilayer cartoon in case any of you were confused by my earlier statements. When the channel is closed, the solute, typically ions, cannot move. When the channel undergoes a conformational change, ions can move according to their concentration gradient. They don't go up the concentration gradient, they go down the concentration gradient. This cartoon has a mistake-ish in it. It suggests that the ions simply pass through the channel without interacting with the protein. That's wrong. The ions will bind to sites within the protein, pretty much just like they bind here. There are binding sites, but the movements through these binding sites are very fast, and many ions can move per conformational change. So when the channel opens, hundreds or even thousands of ions can move. And then the channel might undergo a conformational change to the closed condition. Questions about, well, so here is a cartoon of a passive, so this is the image from your Cell Phys textbook. Here is the image of the channel from your AMP textbook, not much better. Here is an image of a uniporter from your AMP textbook. Again, binding, conformational change, unbinding, conformational change, binding, etc. We're going to need to use, this isn't just random information. You will be using this information about passive transport, well, simple diffusion when we talk about the lungs, passive transport when we talk about the digestive tract and the kidneys. So I'm going to be building on this basic cell physiology when we get to those organ systems big time. That's why this is worth repeating. Questions? So I want to make a point here that in your textbook and in most of the slides I show, proteins are blobs, right? This is a globular diagram of a protein. You know that proteins are not blobs. Proteins have primary, secondary, tertiary, and quaternary structure. You know that from CellPhys. You know that from the poster project that you did in CellPhys. So here is a ribbon diagram of a channel, and here is a ribbon diagram of a uniporter. And so even though we are going to be seeing them like this and like this a lot, you should remember in your brain that they really look more like this. Apply your CellPhys knowledge, maybe even your biochemistry knowledge. I won't be testing you on, I won't ask you to be able to identify that this is a channel and that this is a transporter. That takes years of, I can look at these things and say, yeah, I see that. I'm not going to do that. But I want you to remember that proteins look more like this and less like this. Okay? Let's take a break now for about 5 minutes, maybe 7 minutes, during which I want you to write down some similarities and differences between these passive transport processes, facilitated diffusion or uniport and ion channels. I want you to be able to write it down. So feel free to take a break, step outside and air your face out if you want, but also work on this writing assignment.